Hi, everybody. Can you see the slides and hear me?
Good morning, sir.
Good morning.
Yes,
Good morning, sir.
Okay, good morning. Let's get started. We have a few people less today. I don't know whether it's because of the third wave. A lot of people are infected. Let's see. I think we'll get started. Okay. Instead of doing all the general virology together, I'm just going to try and shuffle a little bit of general virology and specific viruses, mix and match them so that you don't get bored with one or the other. So what we're going to do today, last class we looked at virus structure. Today we're going to look at a group of viruses called picorna viruses. Anybody wants to guess what this term may mean? I'm going to highlight some things to give you a clue. Okay. Does anybody want to guess?
So they are very small
Genetically, it's
range of
RNA.
picometer, and they generally have RNA as the other guys.
Perfect. They're among the smallest RNA viruses. That's why they're called pico RNA viruses or picorna viruses. By a large family of viruses, I mean there are several members in this group of viruses, but they have the smallest RNA virus genomes that are known to infect humans belong to this group of viruses called picorna viruses. Among them, you have several groups called enteroviruses, rhinoviruses, hepatoviruses, and paraicoviruses. Now, at least three of these I think, are decipherable from their names. What do you think enteroviruses mean?
That infect our intestine,
That's correct.
largest.
They have something to do with the gastrointestinal tract, which means entero, right? What do you think rhinoviruses do?
The nasal cavity.
That's right, nose. Rhino means nose. Hepatoviruses,
Liver.
they affect the liver. I'll tell you what echo or para-echo stands for in a bit, but you can decipher what these cause just by their names. Sorry about that. Just doing classes from home sometimes is a challenge. Okay, let's move on. So, what about enteroviruses? Poliovirus is an enterovirus, and they are among the most well-studied human pathogens because they cause something that was debilitating to humans. And so a lot of money was poured into poliovirus research almost about 100 years ago. And now we have almost won the war on poliomyelitis. Okay. What about enteroviruses or what about polioviruses? They are small viruses, like I told you. Their size is also among the at the smaller end. Not only their genome size, the virus size is also at the smaller end. The genome size is only 7 kilobases, which is among the lowest end of RNA viruses. The virus size is also around 25 to 30 nanometers, which is again at the smaller end of virus size range, which ranges from somewhere from 350 nanometers to about 20 nanometers. That's the size range of viruses. So they are at the lower end of the size as well. Now, this is something that you want to remember. It is a common theme that recurs among any virus infections or any viruses that you speak about. The capsid proteins, or the proteins that make up the protective coat for the virus, they are usually numbered or labeled as VP1, 2, 3, 4, 5, 6, and so on for each virus. Now, VP stands for virus protein 1, virus protein 2, virus protein 3, etc. And typically, capsid proteins of all viruses are labeled like this. They're just called virus protein 1, 2, 3, 4, and so on. So that's just a common theme that will recur among all viruses. Now, what are some general properties of enteroviruses? They are non-enveloped viruses, okay, and they are stable in acid pH of the stomach. I'll tell you why these things are important in a bit. They can be isolated from the throat, intestines, and they are shed in feces. Again, all of these things have a certain kind of importance in the pathogenesis of polioviruses. We'll talk more about it by the end of today's class. Sometimes polioviruses or enteroviruses can cause what is referred to as silent infections. Now, by silent infections, what do we mean by silent infections? They are mildly symptomatic or asymptomatic, that you won't even be able to recognize any specific symptom. So they just pass off sometimes without the infected person even knowing that they got poliovirus. That's called silent infection. However, if they only cause silent infections, we wouldn't be reading about them. We are reading about them, and there was so much of investment of money and vaccination. All these things happened because polioviruses or some enteroviruses can cause serious illness. I'll tell you what that is in a bit. They can also grow in cell culture, and some of these viruses may cause cytopathogenic effects. Some may not cause cytopathogenic effect. Now I'm going to tell you a little bit more about the last line that I have here in the slide. First thing that all of you want to understand is that if there are 1,000 viruses that infect humans, just an example, majority of them you can't easily grow in the laboratory or tissue culture because they best grow inside the body, or where there is a 3D architecture that supports the growth of viruses better than tissue culture. However, we managed to learn to grow a large number of viruses in cell culture in the laboratory for diagnostic purposes, for research, for producing vaccines. Humans and scientists have learned to grow a good number of viruses in the laboratory. So enteroviruses are a group of viruses that easily grow in cell culture or tissue culture, which means if you take human cells of a certain kind in a petri dish or a flask and add a sample that contains enteroviruses, these viruses will infect the cells in the petri dish, replicate inside the cells, and when they replicate, they may cause something called cytopathogenic effect or cytopathic effect. Both the terms are used interchangeably, which means they cause tangible damage or changes in the morphology of the cells that they grow in. Anybody has any questions until this point of time? Are you guys with me?
Yes, sir. Yes.
Okay. Okay, so polioviruses or enteroviruses, they can easily grow in cell culture. Some of them produce cytopathic effects. Some of them do not produce cytopathic effect. What does that mean? Many of them, or almost all of them, or most of them, can easily grow in cell culture, but some of them produce tangible morphological changes to the cell culture, while others don't. Okay, that's what this means, with or without CPE. CPE is again another term that is, anytime anybody talks about growing viruses in cell culture, they will talk about this term called cytopathic effect or cytopathogenic effect. The reason being the kind of changes that each kind of virus or each virus produces on a certain kind of cell may be different, and they can help the scientist or the virologist identify or putatively identify the virus. And that's why this term CPE is important, because many viruses cause characteristic CPE or a specific kind of cytopathogenic effect that you can look under the microscope and say, probably this is this virus. Okay, so that's why this term is also, again, will be a recurring term, at least when we talk about cell culture. Now, that's the introduction to enteroviruses. Now, again, this slide, don't be threatened by this slide. Just, you don't have to by heart it. I'll tell you what serotypes are in a bit. I'll tell you what serotypes are in the next couple of slides. But you don't have to, again, by heart these numbers. Enteroviruses include polioviruses. There are another two viruses called Coxsackie A and B virus. Coxsackie is a suburb in New York, and that's where this virus was first isolated, and that's the naming part of Coxsackie. And then there are echoviruses. Remember when we looked at this slide, I told you I'll tell you what echo is in a bit. So what does echo mean? Why were these viruses named as echoviruses? They were named as echoviruses because they were enteric, which means they could infect the intestine and you could isolate them from feces. They were cytopathic, which means they caused CPE when they grew in cell culture. They were human in origin because they were isolated from human feces. But then they were called orphan viruses at that point of time when they were named, which is almost like 75, 80 years ago. They were called echoviruses because people thought they were orphan viruses. What is the meaning of this orphan viruses? So they thought these are enteric viruses. They are isolated from human feces. They grow in cell culture and cause a characteristic change in the morphology of cells. They're cytopathic, but they were not associated with any disease at that point of time when they first named this viruses. So they called orphan because they were not associated with disease. This word orphan is because of lack of association of this virus with any particular disease at that point of time. So they were called echoviruses. Okay, so you don't have to by heart these at all. We'll read a little bit about these later in this course in the next few lectures, but today and the next one or two classes we'll focus on poliovirus. Again, you don't have to by heart this part, don't worry about it. Now there's just one note here. What was referred to as enterovirus 72 several decades ago is now being reclassified as hepatitis A virus. It's just for information's sake, don't worry about it. Now, for any virus, it's important to understand pathogenesis. And pathogenesis does not mean just the disease a virus causes. It also starts, where does pathogenesis start? Anybody wants to answer that? Guys, when we talk about pathogenesis, let's say when we talk about how SARS-CoV-2 causes disease in humans, where do we start? We start with how it gets transmitted. That's a good starting point. Where it replicates, what it causes, and what are some short-term and long-term sequelae, and how does the immune system counter it? All these things are part of the pathogenesis. How long do we continue to shed the virus? All these things are built into this word referred to as pathogenesis. Now, anybody wants to make a guess about how polioviruses are transmitted? Guys, based on whatever I told you, these are enteroviruses, they replicate in the gut. So how do you think they would be transmitted?
So food and
Say
water.
that food and water. Okay. Okay, that's more or less correct answer. So this route, for those of you who are not familiar with this term, I'll explain this term to you. It's most diarrhea causing viruses, and pretty much all the enteroviruses enter through this route called the fecal-oral route. Now, why is it called the fecal-oral route? I'm going to explain that to you. So an infected individual will shed the virus in his or her feces, and then that can contaminate hands, and that can contaminate food or water. And when we eat this food or water, it enters our body. The pathogen enters our body, and this route is called the fecal-oral route. All of you can understand the meaning of fecal-oral. It's shed in the feces, enters through the oral route called the fecal-oral route, and poliovirus is a virus that enters through the fecal-oral route. Now, what happens when the virus enters the body? It enters the mouth, and then it URT stands for upper respiratory tract. So it replicates a little bit in the oropharynx and tonsils. Now, how many of you know what the pharynx is? Where is the pharynx? Guys?
So pharynx is
In
a
the neck
very
region?
Say that again.
In the neck
Pharynx.
region?
Okay, in the neck region, somewhere around the throat, the pharynx is divided. A part of the throat closer to the nose is called nasopharynx. Near the mouth or the opening of the mouth, at the back end of the mouth, that's called the oropharynx. And there's a part below that called the hypopharynx. We don't need to know all that, but this is a part of your throat. That's the important part. Now, tonsils, everybody knows. What are tonsils? Guys, all of you know what tonsils are.
These are the lymphoid tissue present in Peyer's patches.
They are not present in Peyer's patches, but they are lymphoid tissue. That is correct. Peyer's patches is somewhere in the stomach or in the intestine. That's where Peyer's patches is. But tonsils, you're right. You know, on both the sides of your throat, you have this little pink swelling, and that's a collection of immune cells that helps us fight infection. If, for example, if we get exposed to a pathogen in the throat, they are the ones that can immediately fight it, trap it, present antigens, all these things. They are a collection of immune cells. Let's just keep it simple. And they are present on both sides of the throat, and most of us know it. So the virus enters fecal-orally and replicates a little bit in the throat and the tonsils because they have the receptor for this virus, these cells. And then once it replicates a little bit, the virus goes into the stomach and the intestine, where it replicates a little bit more. And this is the Peyer's patches part of poliovirus. I thought I had it in some slide. Maybe we'll see. I think it's there in a slide. But it replicates then a little bit in the intestine. And then once it reaches the intestine, it can spill over into the circulation by means of small capillaries. It enters them and it spills over into the bloodstream. And when virus spills over into the bloodstream for the first time, it's called primary viremia or just viremia. Virus in the blood, emia, anything to do with emia is something to do with the blood. So this will be again, anemia is something to do with lower number of RBCs or hemoglobin in the blood. Just like that, viremia means virus in the blood. Okay. And at this time, the person is asymptomatic and has little virus in the blood. This is not much virus. There's only a small amount of virus in the blood, but there is virus in the blood. So what happens then? So the virus in the blood, because blood goes to all the parts of the body, it has the ability to go and seed other parts of the body which have receptors to this virus. Now, what is the receptor to this virus? It's a surface molecule called CD155. Again, not in the slides because you don't have to necessarily by heart it. I don't think it's in the slides. And you don't have to by heart it. So what are surface molecules? So let's say this is a cell, the nucleus. There are several small proteins in different shapes and sizes that are there on the cell surface, and these are called surface molecules or surface proteins of the cell. And pretty much all the cells have hundreds of different types of surface proteins, all our cells. And CD55 is a surface protein that is associated with the immune system. Is there in several of the immune cells. It also happens to be accidentally expressed by this group of cells called the anterior horn cells. Now, give me a second. Let me try to erase everything and start over again. Now, what are the anterior horn cells? of the spinal cord. All of you know the spinal cord, right? It is how the nerves go from the brain along inside the vertebrae of the backbone. There is a bunch of nerves that go through the vertebrae, and that's called the spinal cord, and all of you know this. Now, tell you a little bit more about the spinal cord that many of you may not know. Let me see what I can do. Okay. Okay. Give me just a second. I'm just going to try and draw something here for you guys. Okay. So now if you take a cross section of the spinal cord, if the spinal cord is running like this, if you were to cut it like this and take a look, this is how it will look. And it has this part right here. This part. Oh, I'm so sorry. This is becoming dirty with this. With the mouse, I don't have good control. Maybe I'll just do it very quickly. Give me a second. Okay, so you have this region. You see this, this, and this. So they are like horns, right? Horns of a bull. So they're called horns. That's the reason for the terminology horns, because they look like horns of the bull. And I'll introduce a small bit of anatomy that many of you may already know. So let's assume that the person is facing this side, okay? Head and eyes facing this side. Okay, this is this side as his eyes and head and all. They're facing like that. This is his back side. Now, and you're just looking at a cross section of his spinal cord. So because this is the front side or the anterior side, and these horns right here, what I'm circling here and here, they are called anterior horns, and these thinner ones at the other end, they're called posterior because they are at the back side of the body or back side of the cross section. They're called posterior horns. Now, these anterior horn cells, what I'm circling in red right now, they are the ones which express CD155 or the receptor for poliovirus. There are other neuronal cells that may express this at lower quantities, but these are the cells that express quite a bit of the receptor for poliovirus. And poliovirus is because it has these cells have the receptor, the virus spills over from the blood, it crosses the blood-brain barrier and goes and infects these cells called the anterior horn cells of the spinal cord. Now, this is just an overview. Again, now for this, at this point, don't worry about this part. Just forget about this part right now. Just focus on this part. Now, let's see how it goes. Fecal-oral route. Very rarely it can be when when a child has it in the throat, when the child sneezes or something, it doesn't induce a cold or anything, but accidental sneezing or coughing can also lead to aerosol mediated ingestion, which is much less common. Most often it is by contaminated food or water, that is called ingestion. Then it replicates a little bit in your tonsils and your oropharynx, that's the back of your mouth or throat. That's where it replicates a little bit. Then it goes through the esophagus, goes to the stomach, and the green part is your intestine, small intestine, where there's something called Peyer's patches. Peyer's patches, again, nothing but a collection of immune cells throughout our body, including our brain, skin, everywhere we have collection of immune cells that help us fight infections locally in different parts of the body, and Peyer's patches are part of it, which contains collection of immune cells somewhere in the small intestine. So that's where it replicates again, and then it spills over into the blood, and then it leads to what is referred to as primary viremia. The virus enters the blood, and then it can seed other organs like brain and other organs, and then there can be what is referred to as secondary viremia when the virus spills over into the blood after replicating in other organs. That's called secondary viremia, and then it can affect the brain or other cells, where Give me a second. Give me a second. Sorry. Sorry about that. Okay. So then it can go and seed the anterior horn cells of the brain, and then it can cause what polioviruses cause, the serious disease, which is paralysis. We'll talk more about it. Now, I told you I'll tell you what serotypes are in a bit. And this is where I'll have to tell you what serotypes are. Now, poliovirus has 3 serotypes, 1, 2, and 3. What do serotypes mean? Okay, now this is poliovirus 1, poliovirus 2, and poliovirus 3. Okay, 1, 2, 3. So what this means is, let's say I get infected with poliovirus 1. I have antibodies. I have my body responded well. I cleared the virus and I have antibodies. Let's say now I get exposed to poliovirus 2. Will my antibodies neutralize or counter this virus? No, they will not. So that is called a different serotype, which means they are so different in terms of the proteins that they express that the antibody response that they elicit is also different, and usually they may not offer cross protection. They may not offer cross protection. That may vary from virus to virus, but the most important thing is the proteins of these serotypes, expressed virus proteins, are so different that they elicit a very different kind of antibody response from the host. And they also don't have any common antigen, which means although all the 3 are polioviruses, they are so different that even the sequence of the amino acid or nucleotides is almost 50% different. So every second nucleotide is different between these serotypes for some proteins, so making them look like almost 3 different viruses which cause the same thing. There is no cross protection among these viruses. Again, when we say serotype, it only means that the virus proteins are different, leading to different kind of antibodies in the infected host. They may be cross-protective, they may not be cross-protective, depending on the virus. In poliovirus, there is little or no cross-protection, which means if I get infected with serotype 1, I can later be infected with 2, and then after that with 3 also, because they almost behave like 3 different viruses. So that's serotype. Everybody with me on serotypes? Anybody has any questions on serotypes?
No such
No,
thing.
sir, I had
Go ahead.
a doubt. Like, what's the difference between a different strain and a serotype?
Okay, so I have some slides on that. Okay, let me put it this way. Changes in the genetic sequence of a virus may or may not result in a different serotype, which means a very small change in the genetic sequence may not lead to a different serotype, but all serotypes have differences in genetic sequence. Does that answer your question, Grima?
Yes, sir.
Okay, I'll tell you more about serotypes and like these terms, variants of concern and all these things. I'll tell you a little bit more. But basically what they mean is that the protein sequence is different, which means obviously the nucleotide sequence is different. Because the proteins, amino acids are different, they elicit a different immune response from the host. That is what sero means. Serology. Serology means antibodies. So they elicit a different kind of antibody response in the host, of course, owing to the differences in genetics, but then all genetic changes may not lead to different serotypes, but that part we'll discuss in a separate class. Okay, does that answer your question for now at least?
Yes, sir.
Okay, okay, so humans are the only susceptible host to poliovirus. That is a very important thing. We'll discuss that, why that's important when we talk about vaccines. Okay. So now polioviruses, there've been a global vaccination, very extensive, and it's been eradicated from most parts of the world except for 1 or 2 countries in Asia and a few more countries in Africa. They've been eradicated from the face of the earth. Now, what about poliovirus pathogenesis? When somebody gets infected, let's assume I get infected, what happens to me when I get infected with poliovirus? So the first thing that you'll need to understand is this term called incubation period. Now, what does incubation period mean? So when somebody gets infected, let's say I get infected today, till about 7 days, I may not show any specific symptoms for poliovirus infection. And that's why the incubation period is somewhere between 1 to 2 weeks for poliovirus. Incubation period is a period when somebody is actually infected, but does not have specific symptoms for the given virus. So that's called the incubation period. When we have the virus is replicating inside our bodies, but we don't necessarily have the symptoms. So that's the incubation period. Now, told you it's the fecal-oral route. It multiplies in the tonsils, it multiplies in the Peyer's patches of the small intestine, spills over into the blood, causes what is referred to as viremia. And then from there, it can seed other organs, other immune cells, and more viremia. And then when there's enough virus, it can cross over to the brain, and infect the anterior horn cells of the spinal cord. Now, only a minority of cases have severe infection of the neurons or the spinal cord. Majority of cases escape it. And CD155 is the receptor, like I already told you. What is the receptor? Now, let's say somebody gets infected with poliovirus. There are 3 possible outcomes. And the first one, which happens more often than not, is a good one, which means it's called subclinical infection. What does this mean? It means that they may have mild or no symptoms, and the body is able to fight off the infection, and they are just fine. No long-term effects. The second one, which happens in a minority of individuals, about let's say 5 to 10% of individuals approximately, they have fever, they have body pain, and a few things, and they may also have something called aseptic meningitis. Without going too much into the term, does anybody know what is meninges?
Brain membrane.
They are like membranes that cover our brain and spinal cord. They are protective in nature, and any term that ends with itis means inflammation. Okay. Meningitis means inflammation of the layers that cover the brain and the spinal cord. Aseptic means no bacteria. Without bacteria, there is inflammation of the brain covering. That's called aseptic meningitis. Septic means with bacteria. Aseptic means without bacteria. Now, this is also fine. They recover well. They just have fever and a few symptoms, and they have minor infection of the meninges, but they recover well. But this is the group that really suffers. They have something called the major illness. Thankfully, it occurs in a small minority of individuals, and this usually happens like after symptoms begin. In 3-4 days after the illness, they have what is referred to as a major illness. Now, I'll tell you what the major illness is. The major illness is the damage to the anterior horn cells. Depending on how much damage and whether the damage is permanent, that part the part of the body that's supplied by the those anterior horn cells which are damaged, that part becomes paralyzed because those neurons are damaged. They cannot send the signals. These are motor neurons. Like, if all of you can understand, these are motor neurons. What are these motor neurons? They are the neurons that control the muscles of the body. Sensory neurons are the ones that are able to, when you touch something hot or when you taste something, all these things, how do you know it? Because those signals go from your skin or your mouth or your nose to the brain, and they recognize this. Those are sensory neurons. but the ones that control muscular action, or muscles, they are called motor neurons, and motor neurons are the ones that are damaged by poliovirus in the anterior horn cells, and the extent of damage to the motor neuron translates to the extent of paralysis or permanent paralysis or mild impairment that is reversible, depends on how much the motor neuron is damaged. Now, this is something that we didn't talk about until this point. I told you anterior horn cells of the spinal cord can be infected, and that leads to paralysis, usually of the lower limb, because those are the neurons that are affected, and that's called the spinal form of poliomyelitis, leading to paralysis of usually one leg or the lower limb. That's the spinal form. But there is a more severe form of polio called the bulbar form. Now, if you look at the brain, let's say this is the brain. Okay, sorry about all the lines. It's just it goes like this. This is the spinal cord. Okay, I'm just going to shade it like this. This is the spinal cord that goes in. At this point, somewhere here, there is something called the medulla oblongata and pons. That is where the respiratory center is. Now, what is the respiratory center? Is a part of located at the base of your brain, at the back of your neck, somewhere there. And that controls respiration. We don't forget to breathe, right? Everything is automatic because this respiratory center controls it. And how does it get the cues? It gets the cues from some sensors for oxygen and CO2 levels that are present in like major arteries and veins. They have sensors, and they give signals to this respiratory center that controls the breathing rate, how deep you breathe, how quickly you breathe. All these things are controlled by this part of the brain at the base of the brain, and that's called the medulla and the pons. And that's where the respiratory center is. And they also may get infected because they also express a certain amount of CD155, the receptor for this virus. And if they get infected, what you have is, instead of paralysis of a lower limb or a leg, you have respiratory paralysis, and this form is much more serious because it can be life-threatening. Because you have respiratory paralysis, basically the respiratory center, the neurons are gone. They don't control the rhythm of your breathing anymore, and they don't tell you to breathe. And breathing is not a voluntary action for the most part, because it happens even when you sleep. It's not really a voluntary action. It's controlled by the center for the most part. So that way, if these neurons are damaged, you have the bulbar form of poliomyelitis, which is much more severe. Now, this cartoon here gives you a summary of what happens in poliomyelitis. This is how you have to break up virus pathogenesis. This is day 0 when the virus enters the body. Like we've discussed in great detail, it replicates a little bit in the throat. Then later in the Peyer's patches, then it spills over in causes viremia. These are the number of days. Okay, within 1 week, there is enough virus in the blood, and that can cross over to the brain. This is the CNS invasion. It can cross over to the brain, and then it can cause what is referred to as a major illness. That can be just meningitis, which is the minor form, or paralysis. And paralysis can be 2 types: paralysis of one lower limb usually, which is the spinal form and the milder form, or paralysis of the respiratory muscles or the neurons, that damage to the neurons that in the respiratory center, and that's called bulbar form of poliomyelitis. Now, prodrome is just non-specific symptoms, minor illness, like again, headache, fever, a few things. They happen in the first few days. Paralysis may happen, like you can see, it requires invasion of the brain. It happens usually after 7 to 14 days, usually at least after a week. Now, if you look at the person's blood, when do they have antibodies? They have antibodies from about 1 week after infection. That's somewhere around this point. They have antibodies. If you look at this whole cartoon, they will continue to have antibodies. If they are paralyzed, they will continue to have paralysis. And this part is again important. Even if they don't have paralysis, or even if they recover very well with a minor illness, they will continue to shed the virus in stool for weeks together. So look at this. This is day 35. That is almost 5 weeks. They still continue to shed the virus. Some of them may continue to shed it for a few months in the stool. So this is again an important aspect. Now, okay, I put this here. Which step in this pathogenesis is crucial for transmission? Anybody wants to answer this from this cartoon? Guys?
So the step that you like mentioned right now.
That's
This
right.
will be.
The shedding in stool is important because it goes, if this does not happen, or this is only for 1 week, there won't be that much of transmission of polio because everything is fecal-oral, and the shedding, replication in the pairs, patches, and shedding in the stool is a critical aspect for the transmission of poliovirus. Now, all of you know it leads to the paralysis of one limb, usually, depending on the anterior horn cells that are affected. And because those motor neurons, when they don't fire, when they stop firing, when they stop sending signal to your muscle, there's something called muscle wasting that happens. The tone of the muscle is lost. I don't know how many of you know, if somebody gets admitted to an ICU for any reason, if they are there for 2 weeks, they may be barely able to stand after the 2 weeks because there is no muscle movement during the 2 weeks. There's something called muscle wasting, which reduces the tone of the muscle and the strength of the muscle significantly because of non-usage. It just becomes withered and thin, and that's why they have very thin. One leg is normal, one leg becomes very thin. That's poliomyelitis. And it is not a disease of this century or anything. This is an Egyptian stele that dates back to 1400 BC, and you can see a withered leg in this individual, which is perfectly, as it is exactly the symptom of poliomyelitis, the spinal form of poliomyelitis. So it's not a new disease. We know that it's been there for a while. Now, what do you think these machines are? These machines are called iron lung machines, and what they do is, as the name suggests, they can alter positive and negative pressure inside this chamber, and they help you breathe when your respiratory center is affected. Now, which form of poliomyelitis would you need this for? The bulbar form, when the respiratory center is affected. Now, in those days, like much before, even when I was a child, much before that, maybe uh, our parents may be more familiar with this, for example. So they used to be in big hospitals. They always had wards which are called iron lung wards. So basically they have this machine, a less sophisticated version of this machines in large numbers, just for patients with bulbar form of poliomyelitis, because it was so rampant before vaccination and it was so life-threatening. They needed these wards. They used to be part of all large hospitals maybe about 60, 70 years ago. They were very common, and slowly with vaccination, they slowly disappeared because it's not needed anymore. Now, we have another 5 minutes or so. Does polio affect adults? Anybody wants to answer that? What do you think? Whatever I showed you, you're all children. Does polio affect adults? Is there any reason it should not? Guys? Nobody wants to answer that? Okay, so yes, it does affect adults. Anybody wants to guess who this is? It was one of the presidents of United States of America, Franklin D. Roosevelt. So he was infected. He was diagnosed with poliomyelitis somewhere, let's say around 40 years of age. He became president much after that, maybe about 10 years after that, he became president. And well, even when he, like from 40 years, he was on a wheelchair because one limb is paralyzed, and he drove this car and he had hand controls for this car. So everything, the brake, the clutch, the gear, everything is controlled by hand, made by Ford for him as the president of America. Because his leg was paralyzed, everything he controlled with a hand. He drove a car completely with hand controls, including the brake and the clutch. So poliomyelitis can affect adults if they have not been exposed to poliovirus as a child. They could still infect adults of any age. Okay, I think that's where we'll stop. Anybody has any questions on today's class? It's almost time, but we have 5 minutes for questions. Any questions in today's class until this point?
Sir,
Yes.
how long does viral shedding last in case of poliovirus?
It goes on for at least like half a dozen weeks, and in some people it has been documented for several months together. They continue to shed, although they may not have any serious symptom.
All right, thank you, sir.
Sure. Anybody else has any other questions? Any part that you did not understand well in today's class?
Sir, I have a question.
Sure.
Like these polioviruses comes under the enteroviruses class, right? But they affect the neural part. Then why it's not categorized as neurovirus or something is written to this?
Okay. It could be. It is a neurotropic virus. There is no doubt about it. But many of these names are derived from several decades ago, when when these viruses were first recognized by scientists, and because they enter through the gastrointestinal tract, they replicated in the stomach, and they were shed in the feces, and they were spread fecal-orally. Scientists at that time called them enteroviruses because they had a lot to do with the GI tract. Although the final disease, everybody understood if you have paralysis, it has nothing to do with GI tract. They named it enteroviruses those days because it had a lot to do in terms of transmission, replication, and shedding. The GI tract was primarily involved. It was named like that. And there are viruses other than poliovirus in this group, which may not necessarily cause paralysis, but what they had in common is this fecal-oral route of entry, replication in the throat, the small intestine, and shedding in the feces. This was common to the other viruses which did not cause paralysis, so they named it enteroviruses. Does that answer your question? Yeah, sure. Anybody else? Any other questions?
Sir,
Yes, sir.
one doubt.
Yeah, sure.
Sir, so you mentioned aseptic meningitis. So
Uh-huh.
bacteria is crossing the blood-brain barrier, right? So how it is without bacteria?
Okay, so this one is a virus, right? You see what I'm saying, Ada?
Okay.
So when you have meningitis, usually the cause is bacterial. Bacteria from your bloodstream crosses over to the meninges or to the crosses the blood-brain barrier and infects the covering of your brain and cause inflammation to the covering of the brain. That's called bacterial meningitis. And or because at that time bacteria is present, that's called septic meningitis. So now, because this is a virus, you have meningitis without any bacteria. You try to culture the CSF for bacteria or look at a gram smear of CSF, cerebrospinal fluid, the fluid that cushions the brain that's present between the meninges. You will not grow any bacteria, and that's why it's called aseptic, but you still have meningitis because it's a virus.
Okay, sir. Thank you.
Does that make sense?
Yeah. Thank you.
Okay, if you have no more questions, I think we can close for today. I've already sent you the slides from last week. At the end of the week, I'll send you slides from every week, and they will have pretty much everything that we discussed in the class. Okay, guys, thank you.
Thank you, sir.
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 Thank you, guys.